All right, let's dive into something that is truly a revolution in medicine. We're talking about a way to program our own cells to hunt down and destroy cancer with, like, surgical precision. It's pretty wild.
Yeah, just think about that for a second. The cure might already be inside of us, just waiting for the right set of instructions. So we're moving beyond searching for some external magic bullet cure. Instead, we're learning to speak the language of our own bodies, the language of DNA, and tell ourselves exactly how to win the fight. Okay, so before we can even begin to rewrite the battle plan, we have to really truly understand the enemy. And in this case, the enemy comes from inside us.
And this is the most crucial point to get. Cancer isn't some foreign invader, like a virus or bacteria. Nope, it's a genetic disease. It happens when our own cells pick up some bad mutations, they stop following the rules, and they just start dividing completely out of control. Essentially, they've gone rogue. This right here shows the massive shift in strategy. For decades, our main weapon was chemotherapy, the sledgehammer. And it's called that for a reason, right? It's basically a poison that kills any fast-growing cell, cancerous or not, which is why the side effects are so horrific. But now, we're moving to immunotherapy. Think of it like a scalpel, a precision tool designed to unleash our own immune system to target just the cancer cells. So how does this whole new approach actually work? Well, it all starts with an army that's already patrolling our bodies every single second of every day. I'm talking about our T cells. Think of these T cells as these highly trained soldiers. They literally go through a kind of boot camp in an organ called the thymus. That's what the T stands for. And in there, they get educated to tell the difference between our healthy cells, our self, and any kind of foreign threat. They are constantly on patrol, always looking for anything that just shouldn't be there. So the first brilliant idea in modern immunotherapy was figuring out that cancer is sneaky. One of its best tricks is to hit the natural brakes on our T cells, basically telling them to stand down. So scientists asked a simple question: what if we just take the brakes off? And that's exactly what checkpoint inhibitors do. They block those brake signals and unleash the full fury of our immune army. And if you want to see the power of this, look no further than President Jimmy Carter. He was facing what was basically a death sentence, melanoma that had spread to his brain. But by using checkpoint inhibitors, they took the brakes off his immune system, and his own T cells went in and cleared the cancer. It was nothing short of miraculous. But here's the thing. Just releasing the brakes isn't always enough. Cancers are clever, and they have other ways to hide. This pushed scientists to ask a revolutionary question. What if we could build a better soldier? And this is the real challenge. Cancer is an evolutionary process. It can change its appearance, almost like putting on a cloak, to make itself invisible to our natural T cells, even when those brakes are off. Our inner army needed a serious technology upgrade to see the unseeable. And that upgrade is CAR T-cell therapy. And this is where it gets truly futuristic. Doctors literally take a patient's own T cells out of their body, they equip them with brand new lab-designed sensors, these are the CARs, and then they infuse them back in as a living drug that's been specifically programmed to hunt that patient's cancer. The story that really woke the world up to this potential was Emily Whitehead's. In 2012, at just 8 years old, she had already exhausted every option for her leukemia. As a last hope, she was enrolled in this experimental therapy. Today, she's not only cured, she's a pre-med student at the University of Pennsylvania. Her story proved to everyone that this was possible. But the tools they used to engineer Emily's cells were, well, they were a bit clunky and not very precise. They were using things like modified viruses to deliver the genetic material, but they couldn't control exactly where in the DNA it went. I mean, it worked, but it wasn't exactly elegant. And then, right at that exact moment in history, a new discovery popped up from the world of basic biology, and it gave us the elegance and precision that was missing. It's called CRISPR Cas-9. And what's so incredible is we didn't invent this in a lab from scratch. We discovered it. It's an ancient immune system that bacteria have been using for eons to fight off viruses. By finding the viral DNA and literally cutting it to pieces, we just figured out how to repurpose it. The system is beautifully simple. It has two parts. First, there's a small guide RNA molecule that acts like a GPS coordinate, finding the exact sequence of DNA you want to edit. Then, that guide brings along the Cas9 protein, which is like a pair of molecular scissors, and it makes a clean cut right at that spot. Find, cut, and then you can edit. And this quote just shows how routine this revolutionary technology has become for researchers. They can literally go online, order a custom guide RNA for any gene they want, and a few days later be editing the source code of a cell in their lab. It's amazing. So now let's see how this incredible precision tool is being used to create the next generation of cancer-fighting T cells. With CRISPR, we've moved way beyond just adding a single new part. We can now act like software developers for our own cells, writing entire programs, long, complex new instructions, and inserting them with surgical precision right into the T cell's main operating system. So what does it actually take to program that perfect killer? How much code do you need to write to make a T cell smart enough and tough enough to defeat some of our deadliest cancers, like solid tumors? Well, the scale is just staggering. This is not a small tweak. In some of the clinical trials going on right now for diseases like prostate cancer, scientists are inserting a program that is 10,000 letters of DNA code long into a patient's T cells. And this isn't just one instruction. It's a whole software package. It includes the CAR receptor to target the cancer, of course, but it also has enhancements to make the T cell more resilient, code to help it survive longer in the body, and new instructions to help it overcome those cloaking mechanisms that cancers use to hide. It is a complete super soldier upgrade. And that just leaves us with this tantalizing question. Now that we have the tools to precisely write the code of life, cancer is just the beginning. The same logic could be applied to autoimmune diseases, to fixing genetic disorders from birth, or maybe one day even tackling the process of aging itself. The instruction manual for our own biology is now wide open.